is called cell mammals. They're now particular. And so here you have between a by 1 centimeter. Now we know cerebellum who complain of things, but this square centimeter square centimeter in a mouse. And mammal the width only so to be something who seem to have more less of these colors. And then how are they to that? It's implemented in 2 speeds. Primarily in the sheet-like architecture. By it's not now on such as so unique to art for some other mammals. So, and of course there are differences, so if you look at, for example, spine density between mouse and humans, they differ. The gene expression cells, like for example, and of these genes. But it's so we think so they actually they're big. Much bigger than one and skip 6 billion. I know. It's here 70%, and there are lots and lots of differences. About. So it's not clear what's the difference that makes it the difference between all of that. Genau faster, but not this difference. Is it the fact that at least that the performance of all systems mouse. Is it a combination? And yeah, I think. All right, so one thing that allow concepts, concept of the constitution, concept of those are sensory context. Percentage of these single neurons. Implants these large-scale electrodes. The patient why and here you can environment in order to actually I have him. Have you ever listened to Neurons? All right, let's see. Sound like any other neuron in any other species. So what you see here is a click is an action potential that we identify using sophisticated signal detection algorithms. These are the of course we only have recordings where it's like Fried and his colleague put the electrodes, so they're in the medial temporal lobe. These structures, because that's where most of the seizures are. And then what we did, this is a long research program over 12 years. I'm just going to summarize it. There's a whole bunch you can read, and there's a whole bunch of articles in Nature. The last two is Neuron and Sounding American. Last year is probably the best. So we discovered things like this. So this is now in the textbook. It's called Jennifer Aniston neuron. What is it? So this is it. So what we show here in general, we show images that we show the patient. So the patient is on the ward, in bed. There's a laptop on their lap, and they're looking at visual pictures. And we're asking them simple questions like, do you know this? Is this a face? Is this a familiar individual? And this is not a tightly controlled psychophysical experiment like we would do with 50 subjects with eye fix and all of that.
You
it's,
say in front of the computer and use the mouse.
Okay, yeah. It's done under clinical conditions. And what you see here, so here at the top you see the individual images that we present for 1 second. And then you can see the response, 6 trials here. These are all randomly intermixed. So you can see the response of 6 trials. Each little tick here is an action potential. And this here is the average over those 6 trials. And the 2 vertical bars denote the image onset and then image offset. So the image stays on there for 1 second. And what you can see here, to most images, it doesn't respond. So these are famous people. So humans, unlike animals, you can't show them just bars or sine wave gratings or random dot, particularly in the medial temporal lobe. Neurons don't respond to that. You have to show them quote meaningful stimuli. What's meaningful stimuli for people are, of course, scenes from movies and celebrities and famous buildings and sites like stuff like that. So here we show Brian Kobe, a famous basketball player, and Pamela Anderson, and a whole bunch of other things, animals here. But then we also have these pictures of this Hollywood actress, Jennifer Aniston. And here you can see very significantly, this neuron responds to these different pictures of Jennifer Aniston. highly significantly, takes 300 milliseconds onset of response. At the time, she was married to some other actor here. And here you see Jennifer Aniston with her then husband, and the neuron didn't respond to that. Right? Who knows why? So these are now known as, because of this, Jennifer Aniston neurons. Here you have another a neuron in a different patient. This is a neuron that responds to Halle Berry, who's one of the Bonds girls, the James Bond's girl. Same thing here. Here it responds to pictures of her, to a cartoon of her. That year, she was the lead in a movie called Catwoman, and the patient had seen the woman, the movie, and so the neuron also responded to her. Now, if you go on the web, as we did, and look for other images of women dressed in leather, there are a lot, then and show, for example, like this, it doesn't respond to them. So in other words, the patient knew this was Halle Berry, and the neuron fired. Here it also fired, of course, you can't do that in an animal. It also responded to the text Halle Berry, but not to other text. So here you have another neuron that responds to the Saddam Hussein, either pictures of him or the text Saddam Hussein or a computer voice saying Saddam Hussein. All right, so here is a neuron that responds to two different things. There's a few messages I really want to leave because people always claim I'm saying this is the grandmother neuron. Here we have a neuron that responds to two stimuli. It mainly responds to the Sydney Opera House that this patient, he was a traveler, had seen and visited. And it also responds to Jennifer Aniston, her husband. The name currently escapes me. All right. You can do lots of things with these neurons, and we've done lots of experiments where we manipulate what the patient sees and then study how that affects the neurons. Here it's a nice experiment, it's sort of a thought control at the level of individual neurons, where the patient looks at a display where we show individual images, and then is asked to change the image by thinking about it. So typically he gets two images of two people, two celebrities superimposed. We record from the two. So what happens? We look for these neurons. Very often, in most sessions, we don't find them. Because if they're there, even if there are 10,000 of them, we're sampling a catch base of probably 500 million neurons. We have a low probability of catching them. But if we do, so let's say there's, in this case was Marilyn Monroe and I forgot, a young American actor. And then we superimpose the pictures and we ask the patient, okay, think of Marilyn Monroe. And then we record from the neuron. If the Marilyn Monroe neuron fires stronger, the image Marilyn Monroe on the screen becomes stronger, more dominant, and the other person fades, and vice versa. So you can see to what extent can the subject learn to control his or her neurons on individual trials just by exerting mental control. And people can. It's quite amazing. You can see the details here. It came out 5 years ago in Nature. So if people do this successfully, and some people do it successfully from the first time they ever tried, which is pretty amazing. I mean, because this is a totally unnatural way of having control over your mental life, right? Because now what you have to do, in fact, we're doing it from 4 neurons, also some distracted neurons. You have to enhance the firing rate of the target neurons. You have to suppress, that's what the evidence shows, simultaneously suppress the firing rate of the distracted neurons. All, of course, you have no idea what's going on, right? Because you don't know about neurons in your head. All you have, you can think about Jennifer Aniston and, Marlene Dietrich, but you don't know about neurons because you don't experience them. So you can do this quite well. You can, as I said, simultaneously enhance some neurons and suppress the other neurons. But interestingly, you can only do this in some parts of the brain. So we only recover in 4. So you can do this in the parahippocampal cortex, in the hippocampus, but you can't do that in the amygdala and entorhinal cortex. I mean, it's not surprising you can't do it everywhere. So for instance, there's no evidence whatsoever we have that you are able to control the neurons that pertain to color. We know in primary visual cortex, in all of your primary visual cortices, the neurons that are specialized that contain color information, so-called parvocellular pathway, and you're unable, if I put a gun to your head and tell you I'm going to shoot you if you see in color, there's nothing you can do about it. So, it's an interesting question. what part of your brain can you control with what other part of your brain. All right. So what we found that there are neurons that are highly selective, that have what's called a selectivity tuning, that are very, very specific. So in other words, of all the thousands or tens of thousands of things I could show you, all the movies that you know, or all the famous actors, or scientists, or doctors, or your friends, or your relatives, a few thousand of them, these neurons only respond to a very small number. It's probably bigger than one, but we only have half an hour time to test it, so we can test all possible people that you know. And we know from doing multiple electrodes, doing it over many years and getting the statistics, there could be on the order of 1 million neurons. So if you think Barack Obama, there could be 1 million neurons, probably less, probably closer to 100,000, but upper bound 10 to the 6 neurons in your middle temporal cortex that specifically respond to Barack Obama, as compared to any president or any black man of that age. And so the conclusion from this is for concepts that you're very familiar with. So for example, we had a young physics student, and we found a neuron that responds very selectively to E equals mc squared. For things that you're very familiar with, whether it's people or your pet dog or your favorite car, or when you're thinking about Huntington disease, there will be neurons. Your brain essentially derives a shorthand. That's the short way of saying it. Your brain derives a shorthand and encapsulates that concept in a very sparse representation that we can tap into. All right. So the general idea to continue this research program, is just the one slide, is to do things like this. So if you're showing things like this to the patient, so fixate here at the cross, and then tell me what you see. Yes? No? Hello? All right, so what you should, so do most of you only see sort of a green disc? Yes. All right, so by the way, anybody who's a naive realist, this shows you how wrong you are. Because what's out there are stationary magenta discs, and there's a hole, right? That's actually present on the screen. What you see is a traveling green disc. Right, so there's a huge discrepancy between what's actually out there and what you perceive in the privacy of your own mind. It's quite remarkable. It's a combination of proximal fading and color after effect. It's a really cute, it's a very powerful illusion. Anybody, I haven't met a single person who doesn't have it. So what you can do now, in principle, using techniques like this, you can record from the brain. You can do single neuron recording and ask, well, at what level of the visual system do the neurons follow the actual physical stimulus, like the photoreceptor? The photoreceptor will follow, depending the short, medium, long wavelength cone photoreceptor, they'll follow very closely the physical stimulus. Somewhere where what's called the neural correlate of consciousness, the NCC, is there will be neurons that only fire for what you see, actually, what you consciously experience. And you can use techniques like this, both in a human and, of course, also in animals to locate that. All right, a lot of fun. So and now we shift from Caltech to the Allen Institute. So I briefly wanted to introduce us. So we are 2 miles down here on the Berg-Gielman. In fact, we're moving this month to a gorgeous new building down in South Lake Union. Some of you may be driven by it. It's 6-story. It's just across from the UW, from the new medical center at Mercer and Westlake. This gorgeous 6-story building that Paul Allen has given us. So we are rapidly expanding. When I came, we are 160. We now double, roughly 320, and we're going to go to 500 by a couple of years from now. And we are not for profit, and we are somewhere a culture between biotech university. We're not a university in the sense we do not have 30 PIs, and every PI can do what they want as long as they can get it funded. We're not that. We're most emphatically not that. Because there are lots of places like UW and like Caltech and Harvard and Stanford and all the other places, and that have that model. So we're trying to do a different model. We're also not a biotech where we have products, and we have to make money. We are funded 90% by Paul Allen. We focus on a few large projects, and we have products. The products is all free and open and public releases of data. That's our. And we use a lot of technology from biotech. So we have program management and we have smart goals and milestones in order to drive that. So we initiated a couple of years ago. Paul signed off on this very large program over 10 years for building cellular level observatories in both the human and mouse cortex. So we're focusing on those two species. And all made possible by this very generous individual who really cares about, he wants to know, he wants to understand how things work. In particular, he wants to understand, because of his background in computers and programming, he wants to understand how does the brain program itself to do everything that we're capable of. Our new building. All right, so there are three words really what we are and how we differ from other institutions here. We're big science, team science, open science. By big science, I mean typically our teams are 30, 50, 60, 100 people. Team science, they're large teams composed from engineering, computer science, anatomists, physiologists, a lot of physicists and computational people to program management. And then we're open science. Everything we do, once it passes internal quality control, goes out on websites where anybody, you can right now download all of our data. You don't have to sign anything. It's all there, together with viewers and dedicated viewers. And if you have dedicated informatic products, and if you have questions, you can send them to us and we'll answer them, including we will rerun experiment if we believe that there's a serious discrepancy between what you think there ought to be and what we have. And so we've done these over the years, these various big data releases. First, of course, the standard Allen, which is now sort of the standard in the field, the Allen mouse atlas, where in 20,000 we expressed all the 20,000 genes throughout the mouse brain using in situ hybridization. We've done the same in the spinal cord, mouse spinal cord. We've done the same in the developing mouse cord. We've done the same in the human, and in a macaque, developing macaque, in human, and in developing human. Yeah, so it's the biggest website, neuroscience website on the internet. We give out 14,000 engineered mice, so we're doing a lot of stuff for the community. And as I said, all the data are accessible versus via APIs as soon as they pass internal quality control. No commercial restrictions. So if you're a big pharma and you make money, good for you. Maybe we hope you acknowledge us. And they are always accessible several years prior to any publication. So all the papers that we publish, these large resource paper in Nature and Nature Neuroscience and Neuron and Cell, they always come out after the actual data is out there, and we've never been scooped. All right, so I just want to focus on the few of the highlights of the human work. So we've done transcriptional atlas, where in 6 postmortem brains that are very carefully selected. We've done transcriptional analysis. And here we see sort of the transcriptional architecture of the adult human brain. Let me explain this, what this is. So here we show, it's a lot of data. We show, you pick 2 different parts of your brain. Let's say the medial, the amygdala and the lateral geniculate nucleus, so the amygdala and primary visual cortex. And you want to know how many genes are differentially expressed. Of the 20,000 or 21,000 genes or so, how many are differentially expressed between any 2 structures. The structures here, this is clickable. You can go on our website and click it and download all the individual units or download all the data if you want. So you can see how homogeneous, genetically speaking, how homogeneously is cortical tissue. Well, and so it's brain tissue, human brain tissue, in 6 neurotypical individuals. So here, for example, what you see, what we knew before, in cerebellum, so this is a log scale. I think the threshold criteria for expression level difference is a factor of 2, I think. And so what this tells you that although, yes, you have 69 billion neurons in your cerebellum, most of which are, of course, are these tiny granular cells. There's actually across the cerebellum, right? Across the cerebellum, if you look at different lobes of it, very, very little difference across all these. I mean, genetically speaking, it's very homogeneous. If you go cortex, within cortex, there are a few special areas, like primary visual cortex is highly specialized, partly because we, of course, we have visual dominant features, we have phobia, etc. And then there is posttemporal gyrus and temporal pole. Again, those are highly specialized, but most of the rest is relatively similar. And where really the differences arise in the striatum and then the pons and in the brainstem. There you see very large differences. But those sheet-like technologies, right, the cerebellum against the sheet and cortex is relatively homogeneous. Of course, there are differences across the cortex, but very interestingly, if you look at the differences, so if you just focus on cortex and say, well, so I sort of computational, I take the tissue and I unroll it. Now I ask, what's the difference in gene expression? It is much smaller than between cortex and other parts of the brain, but there are differences across cortex. And now I ask, if I take two parts of cortex and I ask, how different are the genes? Turns out 50% of that can be explained by the distance between them. In other words, if you sample 2 points on the cortical sheet, the farther they are apart, the more genes they express differentially, which sort of makes sense. And again, it brings home the fact that this is really a physical tissue. It's not an abstract computational thing, it's a physical tissue. And so laws like diffusion laws hold, and you see that reflected in the transcriptional architecture. Yeah, we use different techniques. Let's skip this. We just finished an IV glioblastoma project. Let's skip this. We're doing here with Richard Albogen here TBI-related neurodegeneration. This is the last study I wanted to mention. So as I mentioned, I've worked for many years in the living human brain, recording individual neurons. It's cool, and you can do things like Jennifer Hansen. You can ask people to immediately imagine. Right, so we've done one paper where we asked the subject, well, we just showed you that the picture of a dolphin. Remember that? And if we record from a dolphin, more likely not, the person thinks selectively about dolphin. The dolphin neuron will fire, although there isn't any image. The person's just imagining it. So you can do really cool experiments that are very difficult to do in animals. But of course, you have grave limitation. The patients are very different. The etiology is very different. Genetic background is very different. And most importantly, you don't control where the electrodes are. You have no understanding of both the detailed architecture and also you can't intervene, of course. You cannot do any perturbation experiments, any fine-grained optogenetic, etc. in the human brain. But there is a possibility of taking the human brain out and studying it in the lab. And so we first started this collaboration with Jeff Ogerman. Now, in the meantime, we collaborated with Jeff Ogerman and Andrew Koch. We're setting up a collaboration with Silvergate, and then at the Swedish, we're working with Fredrik Wynn and Charles Koch. So it's a combination of epileptic surgery and tumor surgery. So here, this is a case where the surgeon needs to access a dysfunctional part, deep inside the brain, in the hippocampal area. And so in order to access that, you tunnel through. And so typically it takes out this part of the lateral temporal lobe. And so we've worked out a protocol, and we have a special container with special carboxyl bubbling through it and a whole lot of special stuff to buffer it and protect it. So essentially, we stand outside the surgery room, and we ask the surgeon to cut coagulation, sort of because of the heat, poses problems for us. To cut the tissue off and drop it in our container, and then we have a special car. We drive it over to the lab, and we immediately slice it and dice it. So you get a piece of brain like this here. So on the order of a sugar cube. Sometimes it's smaller. Sometimes it can be much bigger, of course. And then we can then cut that. We can do Nissl. And so now we can do the entire instrumentarium of slice physiology we can apply, but to a human brain. In particular, what we are now getting ready to do, what we call pipelining, where we do each piece of tissue runs through exactly the same process and using under the same standard system, so we can collect this tissue over years and analyze it comprehensively, and not just base conclusion on 10 cells or 100 cells, but on several thousand cells, and make all that material, the morphology, the electrophysiology, the transcription, available to the community at large. So that's what we're now, we've been doing this in this pioneer mode for the last year and a half, And we're now kicking off at the end of this month sort of our formal pipeline where we're doing it on a very highly standardized, rigorous protocol. And so what we're doing this already since 1.5 years in mouse, and we're doing exactly the same protocol, or almost exactly the same protocol in this human tissue. We go in and patch using standard equipment. Right now it's a combination of manual and automatic, but of course we want to move towards automatic, where we do a standard set of electrophysiological protocols, steps, ramps, pulses, pink noise, white noise, frozen noise. In other words, where we have a noise and we repeat it 3 times because we want to study the inherent variability. And we devise this using our model. So we have a variety of computational models that can reproduce the spiking, both abstract, so-called linear integrated fire models, or generalized linear integrated fire models, as well as detailed biophysical models. And so from them, we know what are the best protocols to try. So we get these sort of stimulus. This takes, the minimum protocol takes 25 minutes. Then we repeat it and we do more experiments. And then we inject biocytin, and then we do reconstruction of the dendrites. So here, for example, this is now all human material. You can see, we've done this in a excitatory cell. We've done it in a fast spiking interneuron. This is near infrared. You can see high quality. So our big surprise is, so I've been wondering about this technique since a decade. And everybody who does slices has warned me that it's going to be very, very difficult. Because when people patch in rodents, of course, the vast majority of patching is done in rodents, it's in very young rodents, 10 days, 2 weeks. Because then it's easy to patch, there's no incrustation, no glia, and all of that. And people say if you go even in P56, in 2-month-old mice, it gets already very, very difficult. And so people said, if you're doing it in humans, that would be P10,000, right? If you're 40 years old, 10,000 days, it's going to be impossible. Well, that turned out it's just baloney. It turns out to be relatively easy to record. The cells are actually bigger, that was expected. It's a bigger brain, bigger species. The neurons are bigger. You can record for longer. So we can easily hold cells for up to 2 days, 48 hours. And that's basically doesn't happen in a mouse. After 6 hours, your metabolic gradient runs down. And we know already in monkey it lasts longer. So it may well be that as the longevity of the species goes up, we live obviously much longer than mice, there's more, emphasis on stress, anti-stress mechanism, on oxidative mechanisms, metabolism that prevents the gradients running down. And so we can do beautiful recordings for a long time. So you can see it's biocytin, then we can reconstruct it in 3 dimensions. So now we have a full 3-dimensional model, morphology of it, and then we can use computational biophysical models using neuron to model them. So here you can see on the right, you can see in black is the experimental data. This is for current injection. Here you can see on the extreme right is a single spike in black, the experimental measured one, and then red is the model. So we can do this really pretty well. Because what we want to do, we want to amass sufficiently number of cells so we can model the circuit. Here we've recorded, this may be the first time ever, one of these very rare layer 5 cells these phonoeconomal or Koch cells, or that are that you find in the insula and in part of cingulate cortex. They have this unique morphology, are very big. We can record from them as well. And what we want to drive towards is to try to identify how many different cell types there are. So we know in the retina, this is probably the best explored place anywhere, certainly in the nervous system, where we understand that retina, they aren't just retinal neurons, cones and retinal neurons. We now know they are on the order of 70 types of different neurons. And no retinal physiologist it wouldn't make sense today to just say, well, I recorded from a retinal neuron. You have to say, well, I recorded from a starburst amacrine cell, or I recorded from an alpha-on cell that I know projects to the superior colliculus or to the LGN. Because we understand what happens in the retina because there are these 70 different cell types. They have unique morphologies, they have unique physiology, and we're beginning to understand by taking the individual components out that some are only involved in the melatonin response, some only respond to motion in this direction or that direction, some respond to dimming, etc. So nothing in the retina makes sense outside the light of the cell types. And the big question in the field is, why is this also true for cortex? Is it true that, for instance, we don't know the answer to this question. Is it true that every cell, let's say, or every neuron in the brain falls unambiguously into one cell type in neocortex? We don't know. We've done recent single-cell transcriptional analysis of a couple of thousand cells and find, yes, there seem to be 49 different types of cells, 42 neurons, 42 types of neurons, and 7 types of non-neural cells. But is it true that every cell unambiguously falls into there, or is there a continuum? Can one cell change from one type into another type? Do these cells tile cortex? So we know the retinal neurons tile. In other words, every point in visual space is mapped by at least one of these 70 different cell types. Is that also true in cortex? If I take a pin and put it through cortical sheet, is it true that somewhere within the processing domain, let's say the dendritic tree of every cell type, there will be at least one of these cell types? So these are how different are the cell types between primary visual cortex and secondary visual cortex, between visual cortex and auditory, between sensory cortex and frontal cortex, between a mouse, a monkey, and a human. Those are all questions that we need to answer. But we know at least enough in the visual system. It's absolutely essential to know about cell types. So most people in the field believe it also will be critical to understand the brain in health and in disease, the different cortical cell types. And so that's what we're trying to do. So we're doing this now since 1.5 years already for mouse. So here we have a database. We released it a couple of months ago, where we're systematically in a high-resolution three-dimensional coordinate system that, of course, we published. We do electrophysiology, just like I showed you before in human, but here we're doing it in mice. We're doing biocycling injection. We give those images, put them online. We do the reconstruction. We do computer models, both these abstract computational models, these so-called point neurons, as well as the very detailed biophysical. We are mapping now using RNA-seq techniques, RNA-smarter, V4-seq techniques to look at the level of individual neuron, of the most 8,000 most dominant RNA species, what's actually expressed. And ultimately, of course, we want to use all these different techniques to do a data-driven taxonomy, to understand, yes, they are 47 cell types, and to understand how flexible they are, how much subject are they to plasticity. And so you can go online, or you can see all this data in the mouse. You can click on where it is from V1. You can click on here. You can get the morphology. You can get the electrophysiology. You can get all the modeling parameters. And we're doing the same now in human. And this database in human we're planning to release in 2017. Of course, we also do papers. We do a lot of papers, but the papers come after this work is first done. We are also very interested. We're spending a lot of resources to try to understand the synaptic connections between them. Right now, using a dual patch or a quad patch technique, right, where it's critical. The brain, of course, isn't just a bunch of 86 billion neurons, but they're connected in very specific ways, probably cell type specific ways. So, for example, if it's true that there are 42 neuron types, which is a very satisfactory number for those of us who are nerds, that was a joke. Anyhow, if you don't know what 42 is, it doesn't matter. It's like, so if it's true there are 42 cell types, well, then there's probably one type of connection that connects cell type 27 with cell type 37 that's different in dynamics or plasticity or its weight or its sign between cell type 21 and 38. And so that's 42 squared. So that's, a lot of different cell types. And so here what you see this is again in human. It's all the work that, by the way, done by Ed Lean, who gave a grant round 4 months ago. And this fantastic scientist, Jonathan Ting. Here, Jonathan Ting is recording from 4 neurons in this human slice preparation. And you can see, so here he injects pulses, and these neuron 1, 2, and 3, 1, 2, and 4 aren't connected, but neuron 3 is connected in an inhibitory way to the other 3 neurons, because you can see every time you inject a pulse of current to fire this neuron, you get this nice IPS, you get this current. It's an interneuron. And what you see here, by the way, is very rapid facilitation. So the first pulse gives rise to a large current inflow, but then it rapidly facilitates. here's one that's there as well. So we're trying to do this at scale to try to, as I said, characterize all those hundreds of different cell type specific connections to understand the dynamics of the human circuit in the dish and compared to a mouse. And we're also doing viral techniques. So we're using herpes simplex virus. We're playing on with AAVs and canine associated viruses with other viruses that infect very quickly. Because we have been able to record for 3 days, but the tissue, of course, at some point will also change its characteristics. So we're trying to work with very fast and aggressive viruses. But where we can begin to use, because we don't have transgenic animals, transgenic genes in humans, but using these techniques, we can then couple them with GFP. If we have idea of specific promoter, we could couple them with GFP and then light up. So then we have a slice where certain neurons light up that we know are PV interneurons, for instance. Right now, all we see is sort of we just have to look and try to identify them visually, which is very, very difficult. But of course, if we have promoter, we can couple them to these viruses, then the cells that express the relevant promoter will fluorescent, and then we can home onto that. Here's something, by the way, 5 days in culture from one of these patients. And we've also actually been able, this may be the first time ever, demonstrated optogenetics in human cells. So this is 1 day after infection. It's an HSV. I don't know what the details, what title it is, and what the specifics of HSV is. But here you can see expressed. So it carries a payload with channelrhodopsin coupled to an enhanced yellow fluorescent protein. So we can patch them, and we can pulse light on them, and we can dry them, just like you can do in another. In an animal. So that's really pretty cool. So in principle, these techniques work in humans. All right. So these are plans of what we want to do. Plans in the human space with a separate parallel effort in mouse, where of course we can work in behaving mice. And so we're building these observatories where the mice run in this bank of observatories, and as they run, we have a team of 6 neurosurgeons that these are mouse neurosurgeons. We're not competing. That we put in windows into their skull, and we can record again using transgenic animals. We can record the activity, and sort of we can peer at the mouse brain and decipher using decoding technique what the mouse is thinking as it's seeing visual stimuli. We all do vision. As it's seeing visual stimuli and has to make a decision. We're very interested in collaborating. So we have many of us, including myself, have affiliate appointments with the UW, and we're taking interns. We have some faculty appointments with we have one faculty trying to make more. So we're very interested in working with people here. It's a pretty unique place. It's different than as I said other places, particularly because we have this big science and team science, and comes with a particular type of challenges that's more common in big physics, right? Like at the Large Hadron Collider in Geneva at CERN, where you have, because we have hundreds of people, and the papers that we publish, many of them, carry 100 or 200 authors, and so there's a lot of question: how do you reward people? How do you all make them part of a team? Of course, you want to harness the ambition and the brilliance of the individual scientists, but then you have to time into these larger goals. So it's an interesting challenge. So partly one can think of this as an experiment in the sociology of neuroscience. And once again, it's only possible thanks to generosity and wisdom of Paul Allen. And this is part of our team, our boat. Thank you very much. Yes, question.
Just give us a sense of, so this is very different from the university model. University model spends a lot of time writing grants and all the administrative things like that. What is the time differential? Because you have your own procedural and administrative things. How much time do you save by doing something model rather than?
Well, you don't. There's very little grant writing. In fact, the shoes on the other foot. So I often have to many more no than yes when people ask me they want to write grants, of course, because they're ambitious, they want additional resources, they want to put it on their CV. Most of the time I say no because it doesn't align with our strategic goals. We have a 10-year plan. It's in all the rooms, and so those are the goals we want to achieve. And so if you come and say, well, I want to join you, but then I want to do, let's say, rat development, I say, well, that's very nice, but how does this fit with we have these goals. We promise to Paul, we have an advisory board, they come once or twice a year, look at us, we promise, we committed, we'll try to make these goals. We committed to get the complete cell types. If your grant sort of, well, you get resources, but then they take you off in a different direction, then this is not the right place. We do want people like you to come here and help us because the detailed experiments, I don't know what the best experiment is currently or 2 years from now. So we want people, so the challenge is to tie people in, and there is to give them enough freedom so they can operate in their space and do their own experiment, but in the context of the larger goals. So there's very little administration, there's no teaching, most people, 95% people don't write grants. We have gotten a few big grants because the NIH really likes us. The stresses are more, so you don't have the freedom that you had, like at Caltech, I studied anything I wanted. You don't have that here, but the real stress is that you're part of a team. So people constantly say, well, I just want to quickly program this. But the trouble with doing that quickly, then you leave, and then nobody can read your program anymore, right? This is what happens, of course, in academia. So no, you have to work with we have a central research and a software. They implement it, because then if you leave, we still have a record, and they do it in a systematic way so that the next person who wants it can also use it. And so people have to be, it's common standards, right? So we fought a long time, the EFIS people, the patch people, the 2-photon optical imaging, and their computational people. They all have very different representation of action potentials. Finally, it should all be the same, but people are more likely to use each other's toothbrush. They're more likely to share toothbrush than to share standards. Well, because their advisor used it already, and she got it from her advisor, and they have all this expertise. And so you have to say, well, you have to give a little bit. Yeah, I understand this standard isn't quite what you want, but it's 95%, and the advantage is we can all use the same standard there. So that's where we spend most of the time. On the other hand, we have living examples of particular physics, or Hubble, astronomy and elementary particle physics, where you have thousands of people. The Higgs boson, CMS, and ATLAS, each of those 2 experiments involved 3,000 people. They're mainly academics. So there's a living proof that academic professors, postdoc, and students can do this. But it requires a lot of time spent in talking.
We can't let Patricia ask another question because we just simply don't have time for that. Can I ask a quick question? I was told.
No, it's a very good question. This is what I occupy my time with.
So you showed us this fascinating work that you published at Caltech about consciousness, the aniston neuron. Do you have, the aniston neuron.
Jennifer Aniston.
Do you know where the signal comes from? Obviously, it's visual, but what's the pathway that gets to this? It's not. Do you know?
Okay, so it's not purely visual because it also responds to the spoken name. Some, not all the neurons, but some will respond specifically when the person says Jennifer Aniston. So, we are talking about medial temporal lobe, right? They sit at the output of the entire sensory processing pyramid. We also know that it's top down because, as I mentioned, some of those, we've done one experiment, also published in Nature, where the patient closes her eyes and asks to imagine. And so there, it doesn't come at all through the retina. It's entirely top down. The other thing we know, it's long. The delays are long, surprisingly long, 300 milliseconds, 280, 300, 350 milliseconds to onset of firing. So we know from some tractography work, it's a processing pyramid that goes, down the fusiform gyrus and then goes into entorhinal cortex, perirhinal cortex, into the hippocampus. But it doesn't seem to be direct single pathway, because that would suggest the delay should be much shorter. So it's not quite clear why these delays are so long and whether there's a frontal loop. In fact, these neurons might be closely related to the memory. We have some evidence for that because some neurons learn to respond to the experimentalist. So the patient obviously doesn't know the experimentalist at first, but then for 4 days you live with the patient, And there are some neurons that we find that respond after a while to the experimentalist. So that suggests it's maybe even more closely tied into actual memory. Oh yeah, that's Jennifer Anderson. I know her and I have this rush of association with Jennifer Anderson. It's difficult to do an FMI because they're far too sparse to pick them up using FMIs, you know.
So if I understand you correctly, there's sort of a population of neurons that respond to here, or just think about the concept of fantasy. You guys also looking at, so that seems sort of like a spatial way of thinking about this. You guys also look at the temporal aspect of it, as in if you take 10 neurons and you fire it into different temporal patterns, it seems to be a way to develop.
Yes, so that's always the argument against these very simple types of code, because well, computationally they're just not very efficient. Okay, so the firing rate of these neurons is very low. So typically they fire, if they fire, most neurons don't fire. They have some long tail distribution where the mode may be at 1 or 2 hertz. Most neurons don't fire, they fire at 0.1 hertz. So, and you have 100 or 2 or 300 milliseconds. So there aren't a lot of spikes to do 40 hertz or anything like that with. And we don't, given that sparse number and given the fact we only have 100 electrodes, we cannot make any claims about temporal synchrony, because of course, I'd love to know, let's see, all the 10,000 neurons that are activated, Jennifer Anderson, are they all activated within 2 milliseconds of each other? I can't tell you that. And again, EG is far too crude, because, that they even if there are 50,000 cells among a population of, 500 million, that's a, a dilution of 100,000. All right.
I think that was the fading experiment described when people were asked to think about one of the images. I guess these were all individuals that had a neuron that responded invariably to 2 different stimuli.
I mean, that's how we show them lots of images of people they know or movies they like, and then we find a response of neurons, and then we use that image associated with that neuron. That's how we do it.
And so when they suppress, my understanding from the descriptions that they can suppress the response to one of those stimuli.
Selectively, they could if there are 2 neurons that are competing, 2 images. Josh Brolin, that's the actor. So the figure we published in the Nature was Marilyn Monroe, the classical picture of her with the dress standing on the subway grill. And then there was Josh Brolin, they were superimposed. So the image starts off 50%, it's a composite, 50% Josh Brolin and 50% Marilyn Monroe. And then we asked the patient in one trial, now think of, and I can show you the movie, it's pretty cool. Think of Marilyn Monroe. And then if the firing rate increases, the Josh Brolin becomes more transparent and the Marilyn Monroe becomes more visible. And the goal is for the person to have only Marilyn Monroe present, not at all Josh Brolin. And most people can do this after 5, 6, 7, 8 trials. And of course, you ask them, what do they do? And they give you some weird answer, because as I say, you don't know what your brain does for you. So it's a sort of being John Malkovich type of experiment, because your brain is controlling you. One part of your brain is controlling another part of your brain.
You had mentioned that the 6 brains you had looked at for a specific experiment were very carefully selected. What do you mean by that?
Well, it's very difficult to get decent dead brains. You probably know this better than I do. People don't donate anymore. Men hardly donate. Women are more likely to donate. And then we have a large number of exclusionary criteria. We didn't want, there couldn't be any connection to any homicide or anything like that for legal reasons. Then no suicide, no psychiatric diseases, no addiction, no alcohol, no drug experience. We did a whole battery of tox tests to make sure we get, what is a neurotypical brain, right? No evidence of Alzheimer's. So, many of these people, one had a car accident, one had a stroke. We didn't want long agonal, stage either. So we had to select. So it took us quite a while to get these brains.
Just wondering about the impact of, say, an inflammatory process. Say you mentioned stroke and what that would do to, say, gene expression.
It's a very good question. So we looked at that. So we now have more brains, and we've done. There's a large number of genes that we call, it's now coming out next month in Nature Neuroscience, differentially stable. So some genes are consistently, stably expressed across every brain in the same pattern. So they're high here, medium here, and low here, and you find that in all 6 brains. And lots of others aren't. You know, immediate early genes, of course, wouldn't be, right? Because of things you. So that's a problem. We'd love to do this in 10,000 genes, but it's very expensive. I mean, the technique, the slicing, the expression is very expensive. It takes, 30 people in this pipeline, 30, 40 people, and you simply cannot do it in large numbers that you would ideally like to do it. All right, thank you very much for your talks.
Thank you so much.